This is your third and last video on genetic expression. So as I mentioned before, when the mRNA molecule is being read during the translation process, it is going to be reading every 3 nucleotide bases, which is known as a codon. First, it looks for that AUG, which is your start codon. And then once it finds AUG, it's going to read every 3 bases after it. That's 1 codon, 2 codons, 3 codons. Okay. This is the way that your translation is going to occur. Find that AUG and then read every codon after it, every 3 bases. Because there are 4 different nucleotides in RNA, A, U, G, C, right? That means that you have a possible 64 different codons. You can have AUG, GCA, CAG, UAA, UAG, UAU, etc., etc. So when you look at all the possible combinations of those 4 nucleotides, you end up with, as you see here, a total of 64 possible codons. But you don't have 64 amino acids. So I've already told you that each codon will code for an amino acid. But the really cool and interesting thing is we only have 20 amino acids. This means that some of these codons will actually code for, or I should say, some of these amino acids will actually be coded for by more than 1 codon. So an example of this is your stop codon. Your start codon is AUG. There's only 1 codon that codes for the start, AUG. But there are actually 3 codons that code for your stopping. UUA, UGA, or UAG all count as stop codons. So as the mRNA molecule is being read, once UUA, UGA, or UAG is read, immediately translation stops, and that's your termination. It's always going to look for these 3. Another example here is proline. Look at proline right here in the middle. Let me highlight it for you. Proline here has 4 different codons that code for it. So proline can be CCU, CCC, CCA, or CCG. So when the ribosome is reading that mRNA molecule, if it encounters CCU, it will have a tRNA molecule attach proline to that growing polypeptide sequence. If it reads CCC, it also will have a tRNA molecule attach proline to it. If it reads CCA, tRNA or tRNA molecule will attach proline to it. If it reads CCG, tRNA molecule will add proline to it. Now there are, of course, some amino acids that will only have 1 or 2 codons that code for it, but they're going to be amino acids like proline that are going to have more codons coding for it. And this is why you only have 20 amino acids, but 64 different possible codons. And this is known as your genetic code. Those 64 codons that code for your 20 amino acids, so those 64 possible codons that code for your 20 amino acids is your genetic code. All right. Everything so far sounded awesome, didn't it? You go from a DNA molecule to an RNA molecule to a protein to the new genetic expression. But sometimes things can go wrong. So we're going to look at mutations. When transcription or translation goes wrong. So when something goes wrong with genetic expression, this is known as a mutation. And mutations can either be very, very harmful. They can be helpful or beneficial because they can actually lead to evolution, or they can be extremely damaging. So there is a key video that I like to put on for students when we look at natural selection, but I'm just going to give you a little brief synopsis about it. In the deserts, in desert areas throughout New Mexico and Arizona, those very dry desert environments of Southwest United States has a mouse called a rock pocket mouse. Okay, these rock pocket mice have been heavily studied both for genetics and for natural selection for one simple reason. These different populations, because there are many populations of them, are found in areas where there's sandy area and then there's rocky surfaces. These rocky surfaces are black because they were made from lava. This sandy area is tan because, well, sand is tan colored. And what has been discovered, the really cool thing that has been discovered, is that the little mice that live here in this area are tan colored. The little mice that live here on this area are black in color. They're the same species, but the ones who live on the rocks are black. The one who lives on the sand is tan. And that might sound logical to you, right? I mean, if you are trying to hide from predators and not get eaten by that snake or that hawk or that owl or that lizard, then the best way to do it is to camouflage with your environment. If I'm black, I won't be seen against the black rocks. If I'm tan, I won't be seen against the tan sand. Makes perfect sense. So what happened is initially there was only the sandy area, but when the black area came about, when these molten lava rocks started to grow in the area, these little mice that lived here started moving, walking up on the lava. And as they walked up on the lava, a lot of them started to die. But those who were actually black survived well on the lava. So their babies started to have more black. So the genetic expression, the code that leads to black, is a mutation that is non-favorable if you live in the sand, but it's a mutation that is very favorable if you live in the rocky area. So again, tan color is the normal color of the rock pocket mice. Black color is the mutation. But if I am a rock pocket mouse that lives in the sandy area, the mutation of black color will be bad for me. If I live in the rocky color area, then the black mutation is beneficial to me. So this is how mutations are key for evolution. So some mutations are bad, some are good. Most are going to be nothing. So let's look at some of these mutations in detail. Okay, there are two main types of mutation that are going to occur. One of them is called a point mutation. And in point mutations, this is where you're going to have one base either being substituted for another base. Okay, so your base substitution is the first of those point mutations we're going to look at. Here is one base is changed for another. So say you were supposed to have AAC, and instead of having AAC, now you have AAG. The C was substituted for a G. If this substitution makes a different amino acid, codes for a different amino acid, then it is known as a missense mutation. However, if this change actually codes for the same amino acid, remember when we looked at proline, that it actually has 4 different different codons that code for it. So if this point mutation codes for the same amino acid, then it's known as a silent mutation because there'll be no change. Sure, you have a mutation, you have the wrong amino acid, sorry, not the wrong amino acid. You have the wrong nucleotide base there, but because it coded for the same amino acid, the protein is not altered in any way. So it's called a silent mutation because it does not change the protein. The missense mutation, which is where it does code for a different amino acid, then that means that it will change the protein, and if it changes the protein significantly, that can be either very damaging or favorable in terms of evolution. A nonsense mutation is when one of the bases is altered, creating a stop codon. So specifically, when you end up making a stop codon. And why does that matter? Because if this is my mRNA molecule and my protein is supposed to be this long, But as I was reading it, there is a base substitution, right, that created a stop codon right here. Then that means that this is where I stopped making my protein. And now there's no way that that protein is going to be identical to the way it should be. And that could be the difference between you making the enzyme lactase that will help you process lactose and you not making the protein lactase and then therefore becoming lactose intolerant. A frame-shift mutation is the third type of mutation we're going to talk about. And this is where you are either adding or deleting a single base, or I should say inserting or deleting a single base. When you insert or delete a single base, that can alter the reading frame. And here these 4 images are good at showing these types of mutation. So this first one is showing you a DNA code, right? This is your template DNA code. This is your template, as you can see here, okay? And this is going to be your complementary DNA. Your RNA molecule is made from the template, and you can see here that RNA molecule that is made. And then this RNA molecule will actually code for this polypeptide sequence: methylene, proline, tyrosine, and arginine. And then you end up with the stop codon. So you saw first you had the AUG, which was your start codon, and then you had a stop codon. So this polypeptide sequence is met pro through arc. Okay. In a silent mutation, as we said, that's where a base is substituted, but that base substitution codes for the same exact protein. For the same exact amino acid. So even though you have a mutation, your result, your protein at the end, is exactly the same as the protein here. Here you have met pro through arc. Here you have met pro through arc. Exactly the same. In a missense mutation, you created a stop. Not in a missense. In a missense, it's when you have the base substitution that did not code for the same protein. And so you can see that here. And you started out the same, met pro through, but instead of the 4th amino acid being arginine, it is histone, I forget how you say it, which is HIS. And so now this protein won't be exactly the same. And this could be, for instance, the mutation that leads to you having black fur instead of sandy fur for the rock pocket mice. And if you're a rock pocket mouse that lives on the sandy color, that mutation can mean bye-bye mouse, you're going to be eaten by an owl really, really fast. Or if you live on the rocky sand, it means, guess what? I'm going to get to live because black is favorable there. The last type of mutation that we discussed is the nonsense. And this is where the stop codon is added in place of a different one. So when that base substitution occurs, it actually led to a stop codon occurring too fast. So instead of your amino acid being instead of your protein being 4 amino acids long, met pro through arc, now it's only met pro, and it stopped. Right? 4 key amino acids with your base substitutions. Again, these are going to be your 4 types of base substitutions: no mutation, missense, silent, or nonsense. The last 2 that I had talked about were your types of frame shift mutations, in which you can instead have insertion or deletion instead of substitution of those nucleotide bases. So if you had, wrong pen. If you had AAGC, where before you might have had a substitution that could lead to any of those other 4 things, instead now what you have is a deletion. So instead of having your AAGC, you might have a GC because this A was deleted. Or you might have a AG C because now an extra A was added. So it's frame shift because it's actually going to shift the entire sequence of amino acids that is going to be made. So here is an example of 3 frame shift mutations that are going to occur. So here is the DNA template strand, your mRNA molecule, your protein that should be made from it. So the polypeptide sequence here is MET-LIS-B gly. And that's what should be made. Instead, because you have a deletion or insertion that in this case allowed when the entire sequence of the mRNA molecule shifted due to the insertion or deletion, it actually led to stop molecule, and your protein was never made because instead of being MET-LIS-F-GLY, it is now just MET. In this second example, you have a deletion occurring, and that leads to the frame shift creating for a completely completely different amino acids. So again, instead of being MET-LIS-F-GLY, now it's MET-LIS-LU-ALA. And in the third example that is showing you is where the deletion actually caused there to be a completely different one. So MET-F-GLY and then a stop. So instead of being 4 amino acids long, it is only 3 long, and it is completely different protein that was made from it. So your frame shift mutations can be very damaging because they can create many changes to that polypeptide sequence. And this is the end of chapter 14 on genetic expression. Again, reviewing that genetic expression is the making of proteins from DNA. It has 2 steps, transcription and then translation. Transcription is making an mRNA molecule from that original DNA molecule. That mRNA molecule is then modified, will exit the cell, attached to a ribosome, where it will then be read during the process of translation, making a polypeptide sequence. If that polypeptide sequence is destined to stay in the cytosol, the ribosome will stay in the cytosol. If that polypeptide sequence is destined for the endomembrane system or to exit the cell eventually, then that ribosome will attach or dock on the rough endoplasmic reticulum while that protein is being elongated, while that elongation process of translation is occurring. All right. Once that polypeptide sequence has been made, a few changes and modifications have been added to it to convert it from that primary structure all the way to its secondary, tertiary, and or quaternary structure. Your final protein is made. You have finished genetic expression. Unfortunately, as we looked at, mutations can occur, which can mean a substitution in one of the bases or an actual insertion or deletion in one of those bases, and it's important to know how that may affect the protein. Have a great day, everyone.